Formed in 2021, Ayana Bio is a spin-off from synthetic biology company Ginkgo Bioworks. And it specialises in plant cell culture, so growing plant cells in bioreactors rather than whole plants outside in the field, for example. But when does this technology make commercial sense?
So there are a lot of climate change related problems that are starting to crop up, and you're starting to hear news about some of these things, like cacao. It's starting to impact cacao, it's starting to impact coffee, it's starting to impact things like vanilla, but it's also starting to impact some of the medicinal herbs that you'll find in dietary supplement products as well. A lot of these things are, one, you're getting lower amount of, the crops are starting to be a problem. You're not getting as much of a crop. And then there's also problems right now with wildcrafted products. And a lot of wildcrafted products will never translate to cultivated materials. The bioactives just won't reproduce the same way when you translate it to cultivated as a comparison to the wildcrafted material. Wildcrafted will never be a sustainable way of delivering mass market products, right? So, but if you want to deliver on the health benefits associated with those plants, then you have to find a better way to produce them. We can actually very closely mimic wild type material. In agriculture, we could take a plant with plant cell, and we could essentially make it express 10 to 20 times more of the bioactive than you would ever be able to get from something grown in the ground. And that's a key distinction. So you have to look for stuff where the technology will provide a clear and differentiated advantage. There's a lot of things it actually fixes. I mean, there are alternatives like precision fermentation, but that only will produce like one compound. So the problem that plant cell can solve is that it essentially reproduce the full set of bioactives or the full phytocomplex that's found in a particular plant, which you would never be able to do with, say, precision fermentation. And trying to even rely on the production of those bioactives growing stuff in the ground is very unreliable. And this is a tool that basically sort of changes that completely, because we're able to completely control the production of the bioactives, take an uncontrolled, you have uncontrolled on the ag side, we're able to completely control the production of the bioactives. So, I mean, a short way of saying it is that we can grow plants without growing plants in the ground, meaning we are essentially converting plants into single cells. So you're able to actually grow them in reactors or fermenters, similar to other fermentation-related processes. So, I mean, the way that it starts with the plant. I mean, our journey starts with growing plants. So we have to grow plants. Bioactives are not found usually in the whole plant. They're usually expressed in specific tissue types. So you have a leaf, you have a flower, you have root, you have fruit. So there's all these different tissue types. And the bioactive production will be different. Now the genes that encode for the production of those are the same throughout the whole plant, right? When you're trying to get to plant cell, we're essentially starting with what's called plant cell callus. So you're using a wound repair mechanism that's in the plant, and you're creating these callus cultures of essentially clusters of cells, but it's more like tissue culture. You need to translate that into liquid. So you're going to then try to get those callus cells to grow as individual cells in liquid, essentially in liquid media. In doing that, we're also trying to get away from this concept of tissue. So we don't want to think of things in tissue type. So we're trying to de-differentiate away from the idea of this was from a leaf, or this was from the root, or this was from a flower. We're trying to get cells that essentially are just the best representation of the plant that we can then use to really focus on the production of those bioactives. So one of the things is we don't want to rely on a light source anymore. So light is not, having the need for light is not great from a process standpoint, so we have to get away from light. So if you're not going to expose the cells to light because they're being grown in the dark in these reactors, you have to replace the carbon source, which means you have to feed it sugar. So we have to, of course, use different types we're going to experiment with different types of media, growth media. We're going to use different types of sugar. We're going to use different types of food. And we're going to find out ultimately what's going to work best for the production of those cells. Once you get the cells to grow and you've got the normal media in there, After you've got them grown, the next step is to essentially induce the production of the bioactives. So in order to do that, we need to start putting other things in, like stressors. So you're going to add a stressor that's going to trigger a response. You're going to add things called elicitors, and elicitor is also designed to trigger a response within the cell for the production of bioactives. And then you could also use plant hormones. Plant hormones are very common. They're used as part of the plant that's grown in the ground, and we have to, again, use the same sort of plant hormones. So those are probably the best examples of things that we have to use. Plant cells are relatively new, and there's only been probably one good example of a company actually scaling plant cell culture. So it's still in its infancy. We are still in the early stage. We're a 2-year-old startup, so we still have a little ways to go improving that out. But in order to I think the key is really understanding what's necessary to get to scale. And that's ultimately what Ayana's focused on right now. And that also is important for us having a partner like Ginkgo to help us get there, because what you need to do is you need to be able to select. How do you select the cells, and then how do you optimize those cells at the front end before you ever go to scale? And we think that's going to be the critical part. And the only way to do that is by screening a lot of combinations and permutations of different cells under different conditions with different elicitors to figure out which of those is the optimal way to essentially think about those cells going into a process. In order to do that, you need data. And you need to use a fairly rich multi-omic approach to essentially be able to get to that. And that is a significant part of our strategy in getting to scale. There's not a whole lot known about which of the specific genes encode for the production of the specific bioactives. It's an unknown. There's some published data on some of these here or there, but mostly it's an unknown. If you don't understand what's going on within the cell, you really have no way of actually controlling it. So we actually, because of the lack of available information, actually have to do that. We have to do the work to try to figure out which transcripts are being changed when you make a change. Which transcripts translate to which genes. If you can actually understand where the production is coming from, then you have a better way of controlling it when you get to scale. So we haven't been public with everything we've worked on, but we've already launched a few, like echinacea, lemon balm, and sage, but those are only at a very, very small scale. But we wanted to get something at least out into market. But from a development standpoint, the two that we've talked about the most recently are marigold for, ultimately for lutein, and also for saffron. You have to select ingredients that have the best chance of success from a product market fit standpoint. If we're going to succeed, we need to be able to utilize existing CMO capacity that's out there. So we're spending most of our time trying to make sure plant cell will fit existing capabilities, and we are already exploring that. We're having conversations with CMOs that we're developing relationships with about this, and we're developing our processes based at our facility in Boston that are going to be consistent with being able to adapt and fit into existing CMO capabilities, because we need to be able to build a business. I think it's important to build a business before we ever contemplate wanting to ever put steel in the ground and build anything on our own. Will we build someday? I hope we're able to do that, but we're not going to do it unless we have a business to build it on. So in terms of the conversations we've been having with the investment community, plant cell's still new to the conversation, but we're seeing a tremendous amount of interest so far in that. And I think that largely because they're starting to understand this is like one of the next emerging categories within cellular agriculture. So everybody's sort of taking a look at it. The companies I think that are going to be the most successful in de-risking and being able to demonstrate that they're able to, one, be able to develop the plant cells the way they need to be developed, and the next stage being able to manufacture those, are the companies that are probably going to be the most successful, at least from an investment standpoint. We're one of the lucky companies, I guess, that has raised enough money to sort of get to where we need to get to, and we're confident that we're going to be able to demonstrate that we're going to be able to get to scale.